In short, PD-L1 is a very problematic biomarker. You have 4 different antibodies developed by 2 different companies under 4 different sets of staining conditions, read out in 4 different ways. And so it's no surprise that it's a little bit of a mess at the end of the day. There have been attempts to harmonize these. The Blueprint project presented some preliminary data very recently looking at this. All the antibodies other than SP142 seem to stain relatively similarly. Certainly at high level staining there was good concordance between them. SP142 did seem to stain fewer tumor cells and fewer immune cells as well. One thing people have tried is to look at each test the way the other was supposed to be scored. So can we improve test A by also looking at immune cells as was done in test B? And it turns out that works horribly. Each test works best the way it was developed, and harmonization is actually poorer when you try to look at them in alternative ways. So they certainly, when you use them, they should be used as designed. As together as a group, I think that they show some positive predictive value, but fairly impractical negative predictive value. They don't tell you who not to use these drugs for. So what do we do with this in the real world? In the real world, I think if you're looking to use a therapy that has a biomarker indication, you get that biomarker and you use it as indicated. What do those of us in the research world do? I think we should stop trying to torture PD-L1 into confessing something and instead move on to more useful measures. In the colorectal cancers, I think that mismatch repair, and several other cancers that have mismatch repair, that seems to be a much better biomarker. mRNA expression signatures, particularly centering around interferon gamma and what I might call the ripples of gamma, seem to have legitimate promise. And there's some promise for mutational load as being something of a predictor, although if we could refine that a little bit down to mutations that lead to recognizable neoantigens, we might do yet better.